So now that we know what cells are kind of put together like, right, we know that we can separate the components and examine them via different forms of microscopy. We should probably start talking about what makes a cell, right? So we are going to be talking about eukaryotic cells. As I said before, if you want to get into prokaryotic cells and talk about bacteria, that's a course called microbiology, which of course is offered at RCSJ. So the nucleus in cells, in eukaryotic cells, contains most of the DNA in a eukaryotic cell. Not all 100%. Some of the ribosomes have their own, or sorry, some of the mitochondria have their own DNA, but most of the DNA that makes up what the cell can do. And that means whatever proteins that it needs to make are basically captured, those instructions are captured in that DNA molecule. So ribosomes use the information from DNA to make proteins. This is what I had kind of mentioned before, where the DNA, the gene in a DNA strand, that's what makes a specific protein for a cell. So the nucleus contains all of the cell's genes, right? And the genes here are, they're coding for individual proteins that the cell needs and is usually the most conspicuous organelle. So, we're used to seeing there's your cell, right? And it's got that really dark dot in the middle. That's the nucleus, right? And that contains the DNA of the cell, all right? So the nuclear envelope is what makes up the very outer edge of this organelle, and it encloses the nucleus, which separates it from the cytoplasm. So we're talking about it's a very organized sort of system here. So the eukaryotic cellular nucleus, right? So we're talking about, a nucleus in a cell. It's just this thing here in this, very small inset picture. Now, if you look at it in different magnifications, you're going to see, of course, different structures. This is magnified using an electron microscope. And if you look really close with an electron microscope, we're talking unbelievably high magnification, you can see that this little nucleus is full of pores. Okay, you can kind of see them here, but you can really see them here. They almost look like a flowered carpet or something, right? Well, that inset picture here is describing the structure of those, and they are basically little pores inside, or sorry, on the surface rather, of the nucleus, the membrane of the nucleus, right? So what that means is there's probably stuff going in and out of these pores, going into the nucleus and obviously going out, right? It would make sense if this is kind of like the central control center of the cell for that to happen. So what comes out of the nucleus, remember the DNA is held inside. Should probably that's like the perfect color to just be obscured by that color there. Let me just change this really quick. So there is DNA inside the cell that's eventually turned into, through a process called transcription, RNA, messenger RNA to be specific. This RNA then leaves the nucleus and goes out into the still inside the cell, but it goes into a part of the cell called the cytoplasm. Okay, so now basically what we did was we took a direction from DNA, an instruction, in other words, turned it into a form that can leave the nucleus, and then therefore, basically tell the cell what to build. And as I said before, and as I'll keep saying, it's going to build a protein. Okay, so that's really what's going on here. DNA is kept in the nucleus. It makes an RNA version of that DNA strand, and that's sent out into the cell so that it can actually become protein. Okay, so when that RNA comes out of the nucleus, what it's going to do is link up, or kind of like, I guess bond with, for lack of better terms, what are called ribosomes. Okay, so ribosomes are particles made of a special type of RNA called rRNA, or ribosomal RNA, and protein. Okay, protein makes up the structure of almost everything in cells, right? So ribosomes carry out protein synthesis in two locations. So in other words, they're actually putting the proteins together. They're the ones that construct it, right? So that happens in the cytosol, right? This is the liquid on the inside of the cell. And on the outside of what's called the endoplasmic reticulum. So there's your Words with Friends word for today, right? So the ER, or also the nuclear envelope. So that would be basically like the nuclear membrane, right? We call these bound ribosomes because they're attached to a membrane. The free ribosomes are basically just floating around in the cytosol. So what we're going to spend a lot of time talking about here is what's called the endomembrane system. Okay, so some of which I've already discussed, between the nucleus and the endoplasmic reticulum, but we're also going to extend it from there. But all of this protein synthesis, since we're talking about bound ribosomes, right, is dependent on this membrane system being present. So I think that's important. We should really know this. And guys, I got to say, there are a lot of quiz and exam questions that are based on this system. Okay, so the components of the endomembrane system here are as follows. So I'm not going to read this to you. It's more of an outline slide. Okay, but you can refer back to this if you just want to look at that list. All right, so I gotta switch here. So the endomembrane system is made of the nucleus. So I'm just going to put an N here. Okay. The rough endoplasmic reticulum, which you can see here. The smooth endoplasmic reticulum, which is more, further away from the nucleus generally. Okay. The Golgi apparatus. So you can see that this is what we call the cis Golgi and the trans Golgi. This whole thing here is the Golgi apparatus. And then we have transport vesicles. Okay, so these vesicles are kind of like little bubbles of membrane that are inside the cell that contain what the cell just made. Okay, so, keep in mind, we're talking about making proteins here. Proteins are packaged up in the Golgi and then they're sent into these little vesicles for transport. All right, so we'll be going through this step by step, but this is also another good slide to refer to if you're trying to put everything all together in one shot, one nice linear flow. So the endoplasmic reticulum system, this endomembrane system, accounts for more than half of the total membrane in many eukaryotic cells. So, cells aren't just these, bags of water with a nucleus inside. There's a lot of organization, there's a lot of structure inside, and endoplasmic reticulum makes up a good amount of the membrane that is inside of a cell. So the ER membrane, endoplasmic reticulum in other words, is continuous with the nuclear envelope. So in other words, what surrounds the nucleus, and you can see it in that picture way better than I can draw, right? That's the nuclear envelope. There's the DNA inside, right? You have coming out from this and continuous with it, this whole system of membranes that is continuous with that nuclear envelope. Meaning, this membrane is kind of like, can capture the mRNA and therefore proteins can be made because lining this membrane are a lot of times ribosomes. Ribosomes, again, are the protein factories of the cell. So I'm actually going to go down here first, right? Ribosomes bind to and stud the surface of the rough endoplasmic reticulum. That's what produces proteins and more membranes, okay? Phospholipid bilayer, right? Or phospholipid layers, which are distributed by transport vesicles. So those vesicles, those little bubbles, as I had mentioned in the previous slide, are really what are used to move things around inside and sometimes out of the cell, right? So this is the membrane factory for the cell. Now the smooth endoplasmic reticulum, which is actually a little bit further away. So I don't know, maybe I could actually, just change colors here. Um, that smooth endoplasmic reticulum actually has, so that's a little bit further away, but still connected, has a couple of different functions. Number one, it synthesizes lipids. Okay, so this is where fats come from, triglycerides, right, in the cell. It puts them together from the energy that the cell has acquired. Okay, it also metabolizes carbohydrates, right? So in other words, starch to sugar, which I think is important. It stores calcium. That's really specialized for muscle cells, so we'll talk about that a little bit later. And it also detoxifies poison. And I just kind of wanted to spend a second on this. Detoxifying poison, why don't we talk about humans? I think it's probably the easiest way to, refer to this, is a really important thing, right? Think about our ancestors, and, we had to adapt to. You didn't know what was maybe an edible plant or not. Every once in a while, you come up to one like, hey, I wonder if I can eat these berries or something, right? Well, if it was slightly poisonous, you'll feel the effects, but you might not necessarily die from it because your smooth endoplasmic reticulum can detoxify those poisons. Okay, so my question to you is, what organ in your body has cells that has the most extensive smooth ER? And you would answer your liver, okay? So your liver can't live without it, which is why it's called your liver, right? Your liver is extremely loaded with smooth endoplasmic reticulum. The cells are. So, okay, so think about it. What do humans most often, well, toxify, I guess, make their blood toxic with? Right, alcohol. Okay, so alcohol is a toxin. You're not supposed to have it in your bloodstream. It's not good for you, of course. So if you drink a little bit of alcohol, you feel the effects. People get buzzed. If you drink a lot of alcohol, you will pass out. And if you drink really a whole lot of alcohol, you can die from it, right? So obviously, we're talking about a toxin here. Well, your liver, over the course of, an hour per drink, about, right, can detoxify the alcohol that you put into your system, right? So that's a really great question. MCAT people, if you're planning on taking the MCATs or the HESIs or anything that's a medical based test, or even sometimes the biology tests and stuff, they love this question for some reason. Your liver has the most extensive smooth endoplasmic reticulum of any of the cells in your body because they are the ones that are supposed to be detoxifying your blood, right? They take out the alcohol so that you, well, eventually get sober, right? And people that abuse alcohol for a really, really long time stress their liver out so much and basically use up the cells, pretty much their capacity to do this, that they start to get scars on their liver, which we call cirrhosis, right? So this is a very medically relevant sort of thing here. People are constantly putting toxins in their body. Caffeine is a toxin. When you vape, that's a toxin. Nicotine is a toxin. Alcohol, which we mentioned, these are all toxins that need to be taken out of the blood in order for homeostasis to come back, right? So I think that's an important thing to consider, and this is also why a lot of people have liver problems later in life. Okay, so the endoplasmic reticulum, again, is a biosynthetic factory. So what we're talking about, the factory, what's it actually making? Well, it's making proteins, right? That's a big one. It's also making fats. It's also making, or sorry, breaking down fats rather. It's also breaking down carbs. And it's also synthesizing membrane, right? So, this is a really important part of the cell. It allows the cell to be productive, right? So, in other words, what it's going to make to keep itself alive, and sometimes other places in the body it's going to serve, right? It's going to send things out of itself to go to other places in the body. Think about like your pancreas. Your pancreas makes insulin, right, which helps you regulate blood sugar. That insulin is going to go in this little vesicle here, IN, right? Out to the cell membrane edge and go out into the blood so that other cells can use it. Okay, so in multicellular organisms like people, this is a really important process because sometimes the cells that are making the product, they don't even need the product; they send it out for other cells to use, right? So I think that's important. Um, the Golgi apparatus consists of flattened membranous, these little sacs here, which are called cisternae. Now, if you don't know what a cistern is basically a big holding vessel. Like you can hold a lot of water in it, or, I don't know, in breweries, they can put wine or beer in there or whatever. That's what we'll call a cistern. Okay. So the functions of the Golgi apparatus are to modify the products of the ER. I would almost say it refines them, right? So it makes those proteins in that final shape, that final functional shape. And that's what it's saying in bullet point number 2. Many post-translational modifications. So what this means is the process of making a protein from RNA is called translation, right? So after translation, those proteins can still be modified so that they can be functional or more efficient, right? So that's why we're calling this post-translational modifications. It also manufactures certain macromolecules, which is extremely vague, but it can, right? So that would be fats and what you call it, carbs, right? And then it also sorts and packages materials into transport vesicles. So a lot of people call the Golgi apparatus, it's kind of like the UPS store, right? So they're taking these things the cell makes, packaging them up and shipping them out sometimes or around the cell sometimes so that those materials can be used, right? So that's really what the Golgi apparatus does. And as you can see here, the Golgi apparatus, which is right on the outside of that endoplasmic reticulum, has what's called a cis side and a trans side. Okay, so I mean, I know this is kind of cis and trans is kind of like used now for well, all kinds of social sort of identities, I guess, right? So cis means same side as the ER or the nucleus. Trans means opposite side of the where the ER and the nucleus is, right? So in other words, the cis is closest to the nucleus and the trans is farthest away, those two faces, okay? So as that rough endoplasmic reticulum makes these little vesicles, right? These little packaged up, things, they go into the Golgi, they get refined, modified a little bit, and then they are going to keep going, packaged up for shipment either around the cell, somewhere in inside the cell, away from the nucleus, or in another cell entirely, somewhere else in the body. So a lysosome